think personally, I think the pitch or angle between each base pair is useful. Any opinions?
I have some few points here which I found, but maybe we can go through them and see. We have DNA features that usually describes the helical structure. One is the double helix. The second one is the right hand helix, means the right handedness. Then the dimensions, whereby we have the distance between the bases, and then the diameter, and then the turn of the pitch. We have the major and the minor grooves. We have the sugar phosphate backbone. We have the strong covalent bonding because of the phosphodiester bonds. We have inbuilt directionality. We have complementary pairing of DNA bases, the AT and the GC. And we have the holding together of the pair bases by hydrogen bonds and hydrophobic interior and hydrophobic exterior. So maybe looking at this, A is not there. The number of phosphate groups isn't there. But because we have just the sugar phosphate bond describes it, but not the number of the phosphate groups. It's not there.
And what do the rest think? I believe we're 32 in the group. Let's see who's there. Amira, what do you think? Alrighty, okay. Anyone with a different opinion?
I'm supporting based on the characteristics that have been listed. I'm also supporting the choice A.
Okay. Okay, so which one of the following bases is a purine? The xanthine, thymine, cytosine, uracil. We need to know what the pyrimidines are and what the purine bases are. Midines. Yeah.
Yeah. Generally, the pyrimidines are the one aromatic ring, and they are diamine, cytosine, and uracil. But the pyrimidine and the two aromatic rings, the common ones we know are the guanosine and guanine and adenine. But remember, when we come to the issues of metabolism, the formation of uric acid, we have the purine bases or the purine metabolism of xanthine to hypoxanthine. So where that one qualifies the xanthine to be a purine base.
Yeah. Any objections? Anybody, guys? To discussion. Let's move. Okay, so which one of the following is not used in the during the separation or purification of subcellular organelles? There is the different velocity centrifugation, equilibrium gradient centrifugation, subcellular fractionation, and linear velocity centrifugation.
In the last discussion, somebody said that D was the answer. Linear velocity.
But do we know the differences between them? Personally, I don't know the difference. Did they explain the processes?
No, it wasn't explained, but somebody talked about, I think after going via Google, somebody came with a D, but we didn't come up to why are others part of it. We don't see about anything about them.
Okay, on Google, I've just seen for the first one, this one says different velocity centrifugation is a common procedure used to separate organelles and other subcellular particles based on their sedimentation rates. Yeah. So this is to separate organelles. Yeah. Anyone else who knows what equilibrium gradient centrifugation is?
What I'm seeing from Google about it is like it uses the concept of buoyancy to separate molecules in solution by their differences in density. But it hasn't indicated anything about subcellular organelles, but it just uses the concept of buoyancy to separate the molecules in solution by their differences in density.
The subcellular one was for the this one, the different velocity one.
Yeah.
Yeah.
So there is not much difference between the different velocity centrifugation and the subcellular, right?
I shouldn't say, but they could be related.
Because here in Google, I don't see any difference.
Used to isolate early and late endosomes by performing a series of centrifugation steps. That's what subcellular fractionation is. What did you get on Google, Fatma?
Fatma unauliza?
Okay, should we move?
Irregardless, daktari, irregardless.
Okay, so which of the following represents the peptide linkage?
We had answered this. It said the choice B.
D.
Boy.
Boy. Okay.
Yeah.
All the following are essential amino acids except there's valine, arginine, leucine, and proline. I'm thinking proline, but there's an image that was saying arginine is also non-essential, but I'm not too sure. If any one of you has an explanation, please go ahead.
Or even mnemonic for memorizing the essential amino acids or for knowing the non-essential ones.
Could you please type it out in the chat box?
Yeah. Amadivya, do you have one?
I don't have one personally.
There's one already in the chat box.
Yeah, Belinda sent something.
Just copy and paste it. PBT, TIM, Team Home, okay.
So I read somewhere how you'd say it to remember is you'd say private team hall. So the phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine, and lysine.
Thank you. All right, so the mineral associated with cytochrome is copper, magnesium, phosphorus, and iron. So there's a place I saw there's copper and iron, so I don't know. I don't know what you guys would go with.
I thought it's iron, but I'm not sure now.
Sorry, guys, what did you say?
I thought it's iron because the CYP family contains heme as a cofactor, that heme functions as a cofactor for the CYP family. So I thought it's iron. Now I'm not so sure.
Hey, guys, it's a discussion, kindly. Anyone who has a different opinion.
I would also go with iron because iron is the one associated with cytochrome activity.
Okay. Okay, so DNA is composed of repeating units of ribonucleosides, ribonucleotides, deoxyribonucleotides, and deoxyribonucleosides. I think C is the answer. So on the group, someone has said, Frank has said, cytochrome is a general term for a group of proteins that contain a heme group. Okay. Is everyone okay with C? Or number 42?
For number
Yeah.
41, I think it's both iron and copper.
But I feel like here you'll just have to choose the most probable answer. Okay. So someone has
This
sent
iron containing.
is for cytochrome A3. Okay.
Cytochrome A3, it says in Google, it's a terminal electron donor. So cytochrome, it contains cytochrome A3 contains both iron and copper. So what it does, it picks the ions electron and passes through to the copper. So I think most appropriate answer is iron.
Iron, yeah. Okay. Cellulose is made up of branch glucose molecules linked by, I think this was supposed to be a beta, 1,4-glycosidic bond, unbranched glucose molecules linked by beta 1,4-glycosidic, branch glucose molecules linked by 1,6, and unbranched glucose molecules 1,6. Okay, cellulose.
It's unbranched.
It's unbranched.
Yeah, it's unbranched. Yeah, starch and glycogen, they are branched, but cellulose is unbranched.
Fine. Lipids are insoluble in water because they are hydrophobic. Yeah. The membrane proteins that catalyze active transport reactions.
Excuse me.
Yeah.
Just a minute, Divya. I think we was not just answering questions, but we were to discuss more about each of them.
Okay, so
If
feel
I'm not
free to
wrong.
discuss. It's a discussion. I'm not the only one to talk, so I believe everyone can talk.
Yeah, so just don't jump to the other questions. I think we give time for people to talk.
Yeah, well, most of the time it's all silent, so you can feel free to talk now.
So for number 43, we've gone with B. How have we come to 1,4 but not 1,6? It's unbranched, yes, but the linkage, is it 1,4 or 1,6?
Yeah, it's 1,4.
It's
1,4.
yeah.
All right.
On this, Divya, maybe we can check on linkage in starch, in glycogen. We can discuss why we say beta. What's unique in the anomeric carbon that makes one beta, another one alpha. So if someone knows, please just unmute and explain, because it takes
Okay,
time.
I know for the one of beta and alpha. The alpha, I remember the alpha glycosidic linkage is between carbons with the same stereochemistry. And then the beta is with the different carbons of two carbons with different stereochemistry.
So this is in regards to the glycosidic bond.
Yeah.
Okay. So if Divya, can you display a diagram of an alpha glycosidic bond versus one of a beta glycosidic bond, and then we're able to match the explanation to an example, so that we're able to understand it more.
Like the beta, it's directly upwards of the anomeric carbon. And then the alpha, it's below the anomeric carbon. It's down of the anomeric carbon. I think that's what Nani were explaining when we were doing discussion. If I'm not wrong.
Yeah. Because I remember us saying beta up, alpha down.
Alpha down.
Yeah.
So which means beta, it will have different stereochemistry because it's upside down.
Okay.
And then the alpha, it will have the same.
Okay. Then 1,4 like okay. After I go 1, 2, 3, 4. Then for starch and glycogen, because we've seen cellulose, we've seen it's a beta 1,4 glycosidic bond. So what about starch? Because I thought starch, I think it's alpha. Someone can confirm.
Starch is alpha 1,4 glycosidic linkage.
There it is. Yeah, in food chemistry. Just the okay. Just go up, Divya. Yeah, there, the yellow one. The below. Yeah, that one. Yeah, so yeah, cellulose is a beta 1,4 glycosidic bond. starch and glycogen are alpha 1,4, branched glycosidic. Okay. See.
But wait, guys, how do you know which one is unbranched and branched from these?
I'm basing this off what we had said earlier, that cellulose is unbranched, whereas starch and glycogen are branched.
Unbranched.
Yeah, but we can zoom further and see where the branching is occurring.
So you want me to display another one?
Go previous image, then
This one?
click view image at the down. No.
Oh, okay, I've seen it.
Yeah.
Oh,
You viewed the
yeah.
So, can you zoom in now on this particular page?
I'm trying to.
Yeah. That's fine. So, since cellulose. I have not seen where the branching is occurring. Perhaps the branching
different
occurs in
image.
the polymer, or I don't know.
I think the other image was showing the branching clearly than this one. There is another image she wasn't displaying. Yeah, this one. You can see for the continuous chain is 1,4, but the branching is occurring at 1,6.
Yeah, true. Okay. Thank you, Divya.
No worries.
Yeah.
So you say it's starch and glycogen.
NS is requesting that the branching be explained again.
Okay, so I think when you, I don't know, one.
Okay, so Divya, this part is the part. Can you see my cursor? I don't know if you can see it. Which part is showing the branch in this photos?
I'm as equally confused as you are. Because
It's
here,
that 1,6, that alpha 1,6 linkage.
but wait, look, it's you count it from here, right? There's 1, 2. Let me just add it. There's this one will be your 1.
Yeah.
This will be your 2, 3, 4,
5,
and 6.
6 on top. No, 6 is above.
Yeah, the 6 is the CH2.
Yeah.
Oh yeah, shit. Yeah, sorry.
Language, Divya. This is recorded.
Sorry. Sorry. My
Don't
apologies.
worry. It's okay.
So and then this will be, so how does the 4 come?
The next one.
This one will be
No, count the next one, Divya. Yeah, there.
Yeah, this one, right?
No, the other one. The next one.
The
right
one.
No,
yes,
the one on your far right.
On the
lower
right
line.
side. From the one that you've already counted. Lower right side.
Lower right side.
That
here.
one,
Yeah, this one. Yeah, this one.
Name, so this is where
number
this
table. Yeah, that is number 6. Number
okay.
1, 2, 3, 4,
and
5.
5.
Yeah,
That makes
Yeah.
sense. Okay. Samiha, have you understood?
No, I've not understood. It wasn't Samiha, it was me.
Oh, okay, sorry. So 1 is this one.
Okay.
Are you
So
looking
that's
at
the anomeric carbon.
I think for the continuous, the 2 lines, the one with having 2 sugars and the other line having 3 sugars, those are the continuous lines which are not unbranched. But at carbon 1 of the second sugar on top, on the far right top, and the one below is the one which is forming the branching.
Mm.
Yeah. So the branching is occurring at carbon 1 and carbon 6. But for the continuous linkage is carbon 1 and carbon 4.
Okay.
There is someone who sent this. Allow me to display. Yeah. Let me just erase.
Also, unbranched is the linear one. Amma?
So if the peptide is not branched, we will not have this linkage between carbon 1 and carbon 6. We will have only one line of sugars continuously. We will not have 2 lines. Use layman language like of 2 lines, but like here we are having 2 lines, one having 2 sugars and the other one having 4. So the one which will not be branched will have only one continuous line of sugars.
Oh, that makes sense.
That makes sense, yeah.
Yeah. That makes sense. Anyone else who has a question, speak now before I get told. Instead of staying silent. Done? Okay. Shrina, you have anything to say?
Yes, I wanted to talk about I think we could discuss about since we've started talking about starch, we could also remind ourselves about how amylase and what's the other one?
amylopectin. As I was
Yes.
headed there, yeah.
Yes, we could discuss about it because
Yeah.
we've opened this topic.
Oh,
So, sorry?
Nothing.
Divya?
Sorry. Yeah.
Hi. Oh, before we go to starch, was there any other that had a glycosidic that was a 1,6? That was a 1,6 glycosidic bond.
Glycogen. Glycogen, I think, has 1,4 and 1,6. That is why we call it branched, if I'm not wrong.
Thanks.
True.
You're welcome. You're welcome.
Okay, I have an issue sharing with my laptop. Can someone please share?
Anyone who can share?
Yeah, just.
Okay.
But I might have issues like last time, so.
No worries, no worries. We'll manage. We can see your screen, David. We can see your screen.
Okay. We were talking about 1,6.
Yeah. So for the starch, we want to talk about starch.
No, I just somebody was asking which carbohydrate is joined by alpha 1,6 glycol. I think it's what did I say? Glycogen, I think. It has both beta 1, 4 and alpha 1,6.
Yeah. It has both. Glycogen 1,6, and even cellulose also has that alpha 1,6.
But it's unbranched.
Pardon? Sorry.
It is
Starch. Sorry, guy. I confused Nene.
Yeah.
Starch has also that alpha 1,6.
Yeah.
So on starch, okay, what I know is that starch is made up of amylose and amylopectin, but amylopectin is the main part, or rather constitutes like 80% of starch. And now amylopectin is the one that is where branching will be taking place. So it is the branched molecule. Whereas in amylose is just made up of that linear alpha 1 for glycosidic linkages. But the amylopectin is where we'll see that branching at alpha 1,6. Yeah.
Wait, Gaius, you said amylose is the main component.
No, amylopectin. Amylopectin is the main thing. It's the main component, like 80%.
Yes, and also in addition that the amylose is the one which is soluble. The one which has the lesser amount is the one which is soluble. Then amylopectin is not soluble.
Yeah, nice. So amylose in water soluble. Amylopectin is the water insoluble. It's the one that forms the gel.
Yeah.
I think it's the opposite.
No, it's that way. It's that way. I'm here with the textbook. It's that way.
Okay, so how is it?
Amylose is the water soluble portion, and it constitutes about 10 to 20%. Then amylopectin, water insoluble, 80% of starch, and it is branched alpha 1,6 linkages. Then I'm seeing Dr. Sam from Kabarak. I know he has a lot to add on the uses of starch as an excipient. So he can also add on how starch is used as an excipient.
My net is really poor. I don't know if you guys can hear me, but generally we use starch as a disintegrant. So
Nice. We had used some starch is used as a disintegrant. What of as a binder? Can it function as a binder?
it can actually function as a binder as well, and a bulking agent as well.
What about the tests?
The test for starch, let me...
Just the iodine solution.
Yeah.
Turns blue, right?
Yeah. Starch will form that blue complex with iodine.
And then like for the other tests, it would be like for reducing and non-reducing sugars.
Yeah. But now that's for the kina monosaccharides and disaccharides major, whereby
Yeah.
sucrose is the non-reducing one. The other common ones, kina lactose, galactose, they are reducing. Glucose.
Okay, so anyone wants to add more? Divya?
No, I'm good.
Sawa. Lipids are insoluble in water, we said, because they're hydrophobic. Do we want to remind ourselves of anything about the lipids?
I think there's nothing burning there. We can if we see another question that relates to it, we'll revise.
Sawa. The membrane proteins that catalyze active transport react.
What of the classification of lipids?
You can take us through.
Are you talking about the non-saponifiable and saponifiable lipids? Whoever talked about the classification.
I think, yeah, it's more the ones we have the simple lipids, compound lipids, the derived lipids, and the lipids which are complex to other compounds.
Mm-hmm.
Yeah, but okay, for the simple lipids, those ones they are just esters of fatty acids with glycerol. Okay, yeah, in short, here just an ester of a fatty acid with glycerol that constitutes a simple lipid. But now the compound lipids, these are the fatty acids that are esterified with alcohol, but also contain other groups. So based on what group they contain, they can be called phospholipids, which contain that phosphoric acid, or they can be non-phosphorylated. Now for the non-phosphorylated one, we go ahead and find a lot of things like the glycosphingolipids, which are attached to a carbohydrate. Yeah. Then the derived lipids are the ones which are derived from precursors of lipids. A good example is now the fatty acids and the steroids. But honestly, I don't have a summary of classification of lipids. If someone else does have, they can give it, which will be much easier.
Maybe a little addition on the same on the complex lipids. We can have them like the membrane complex lipids, which we have the phospholipids and glycolipids, as you had said earlier. But now looking at the phospholipids, they can either be glycerol phospholipids or sphingophospholipids. And most of these would be like reservoirs on the cell membranes. That's why you would get something like phosphoinositol, like phosphatidylcholine, phosphatidylethanolamine, and the rest. I am not so sure, but they have a function on the cell membrane and also the blood grouping or the typing. And also some of them they have the antigenic variability, which are used either to detect some of the viruses like the cardiolipins. But that will be more on the complex lipids, not the simple lipids.
Okay, so just to add on that, when we're talking about the classification of lipids, so you have fatty acids, which is like saturated and unsaturated, which is this oleic acid and all these other palmitic acid, all those. Then you have non-glyceride lipids, which now includes like waxes, steroids and sphingolipids. And then you have lipoproteins also. That's part of like the complex lipids. And then you have like glycerides. Yeah.
And also something little is to remember that the body is not able to synthesize the fatty acids, which have double bonds. So usually we get them as essential fatty acids, such as linoleic. That's why you would get them from like omega-3, omega-6, because the body is not in a position to synthesize them. It usually synthesizes the ones which are unsaturated, such as palmitic and stearic acid.
Okay, moving on. The membrane proteins that catalyze active transport reactions differ from soluble enzyme in that A, they do not enhance the rate of reaction. Their products of reaction move in a specific direction. The substrates of the reaction are all outside the cell, or they are permanently charged during the reaction.
Let me go with choice A, out of knowing that enzymes are the only stuff that enhance the rate of a reaction. But I don't know if they are permanently charged during the reaction because when we are saying membrane proteins that catalyze active transport, a very good example is the sodium potassium ATPase. So thinking of the sodium potassium ATPase, choice C is out. The substrates of the reactions are all outside the cell. I think we can agree choice C is out. Because sodium is usually inside the cell, potassium outside, and then it pumps sodium out, potassium in. But their products move in a specific direction. Now, I don't know about B and D, but me I'd go with choice A because I don't know if they catalyze that rate.
Are they not moving in counter directions? Not specific direction.
Yeah, that's what I would
That
think
one is
actually.
that one is also out. So I don't know if they're permanently charged, but I do agree with you. Choice A is correct.
Glycolytic pathway regulation involves feedback and inhibition by ATP, allosteric inhibition by ADP, allosteric stimulation by ATP,
Wait,
feedback
there's someone
stimulation.
saying C is the answer regarding the substrates of the reaction are all outside the cell. Okay, never mind. They've saved me.
This normal saline is messing with us. Dr. Patel, you're muted.
Glycolytic pathway regulation. Somebody can take us through the glycolysis pathway. They can give us a summary.
We have Seraphine in the house. I think Seraphine is a big fan of glycolysis. Even her research was on the cofactors of glycolysis. And she discovered 2 new cofactors. I think we can upload her as she prepares to tell us about glycolysis. Okay, it seems she's not around. So for glycolysis, the starting substrate, of course, is glucose. It's phosphorylated to glucose 6-phosphate. And then we know that phosphorylation step is irreversible. And that phosphorylation step is also important because glucose is osmotically active. It can pull water towards the cell. So glucose, once it enters a cell, it has to be phosphorylated. If not, it can end up being toxic to the cell by that osmotic pool of water leading to bursting of the cell. Yeah, so after phosphorylation, I'm not going to look at all the steps, but the rate limiting step is the conversion, or rather phosphorylation of fructose 6-phosphate to fructose 1,6-biphosphate, which is catalyzed by phosphofructokinase, PFK. That is the rate limiting step. And there are 3 key modulators that act on that rate limiting step to alter glycolysis. The first one is, the first two is ATP and citrate. ATP and citrate act allosterically on PFK to inhibit its action, hence inhibiting glycolysis. Then the other one is AMP. AMP still allosterically on PFK to stimulate glycolysis, or to stimulate the phosphofructokinase. Then we go further to just mention that from glycolysis itself, just glycolysis by its own, we make 4 ATPs, but we use 2 ATPs. That means the net yield from glycolysis alone is just 2 ATP molecules. But also in glycolysis, we make 2 NADH molecules. And we know 1 NADH molecule is equals to 2.5 ATPs. That is when the NADH molecule goes to the electron transport chain. So if we are making 2, it will be 2 by 2.5, we get it's 5 plus the 2 net. So glycolysis in total gives us 7 ATPs. That is when you combine glycolysis with the electron transport chain. It gives us a net of 7 ATPs. But glycolysis alone just gives us a net of 2 ATPs. Violet Mamba is in 8 ATPs. Kindly explain why 8. Preparatory phase uses 2.
You see, on preparatory, you use 2 ATP. On the payoff, you generate 6 plus 2 plus 2 makes it 8. You see, on this stage, glyceraldehyde 3-phosphate dehydrogenase, you generate H, which when taken to electron transport, inakuwa 6 ATP. Phosphoglycerate kinase, pyruvate kinase 2 ATP.
Okay, let me share this on the group. There's a biochemistry textbook, I think is the most recent edition. It corrected and said NADH does not give 3 ATPs, hence which will end up giving 6. So 6 plus 4 becomes 10 ATPs, then growth of 10, then net of 8. With more recent research, they found that NADH just gives 2.5 ATPs, not 3. Let me share that reference so that you don't take me on my word. Shrina, I think we can continue. Meanwhile, I'll share a photo on the group so we can continue.
During catabolism, about 40% of the energy from oxidizing glucose is used to synthesize ATP, and the remaining 60% is reduced to NADP, is mainly lost as heat, remains in the product of metabolism, or is stored as fat.
I'd go with it remains in the products of metabolism, because if they are talking about 40% of the energy used to synthesize ATP, essentially what we are talking about in glycolysis is that we use 2 ATPs, then the other ATPs, or we are able to form this kina NADH nini, but I'm not so sure. It can be either C or D, but I'm not so sure.
I agree with you. I don't know if someone else will disagree, but I think it also remains in the product of metabolism.
Me, I would go with B, because during catabolism there is a lot of heat produced. So most of it, a small percentage is used to produce ATP, which is in the form of energy, and a greater percentage is lost as heat.
I'll also go with B, because for the same reason. Think B. What do the others think?
I'll also go with B.
Does anyone want to add something, or we go to the next one? The released energy obtained by oxidation of glucose is stored as CMP, ADP, ATP, or NAD+. I think ATP.
Yeah, I'll also go for ATP.
I would also go with the ATP.
Okay. A kinase is an enzyme that removes phosphate groups of substrate, uses ATP to add phosphate group to the substrate, uses NADH to change the oxidation state of the substrate, or removes water from double bond.
I think at the one for adding phosphate to the substrate.
Yes.
That uses ATP, yeah?
Yeah, uses ATP to add phosphate, yeah.
Yeah.
Because for the removing phosphate, we had seen it phosphatase.
So wait, the answer is B.
Yes.
My thoughts.
Okay, could you remind us the other classes?
Maybe someone else can do that.
Anyone?
Let me see.
Maybe what I can add on is on the classification of enzymes. According to I think it's more of universal, something about chemistry. They are classified into 7 groups. And the first one is oxoreductases, whereby they work by an addition or removal of a nitrogen from a compound or from a molecule. Then we would have transferases. The transferase is where now our kinases comes in because they transfer another molecule apart from hydrogen. Then we have the hydrolysis. The hydrolysis will cleave the molecule with an addition of water. Then we have the lyases. The lyase will cleave the molecule, but no addition of a water molecule. I mean, no, they'll cleave the molecule, but they don't have anything to do with an addition of water molecule. Then we have the lyase, we have the hydrolase, we have the ligases. Ligases, most of these are synthetases, because I think we remember the difference between a synthetase and a synthase. Because this specifically is more of a class of synthetases because they utilize ATP for their mechanism of action, different from the synthases which do not require an ATP. So I hope I've finished the 7. We have the oxidoreductases, we have the transferases, we have the hydrolyses, and we have the lyases and ligases. What is the 7th one? Maybe if we can go through and check.
There's isomerase.
Isomerase is perfect. Thank you. Yeah, isomerase. Sure.
Are they 7 or 6?
I think there are six. Can we check probably? I think there are six
Yeah,
or seven.
yeah, they're usually six.
Six?
They Nini, the isomerase is class 5 because even the number matters. Oxidoreductase is class 1.
True.
Yeah, because the
True. Very
mnemonic, okay, I use OTH lil. So at number 5 is where we should have our isomerase.
thanks.
Yeah. Yeah, so based on this, let's see. Maybe someone can place the other choices in the right, according to the choices, say what class of enzyme the choice is likely describing. Maybe Dr. Kanja Ann. Or anyone else, or anyone else.
Maybe looking at this, I would put a phosphatase removes a phosphatase group will be under the transferases because it will transfer it to another place. Then the use of ATP to add a phosphate group, it will still be under the transferases. But the use of NADH to change the oxidation state, then it will be number 1. And then they removes water from a double bond, then it will be under hydrolysis.
Nice. Thank you so much. I'm seeing Frank has said the 7th class is the translocases. They facilitate the movement of molecules or ions across membranes, the translocases. That's the 7th class.
Thank you, everyone. In the glycogen synthase reactions, the precursor to glycogen is glucose 6-phosphate UTP, glucose UTP, glucose or glucose 6-phosphate. Sorry, the last one is glucose 5.
I think it's UDP glucose.
And the first one would be for the glycolysis pathway, right?
Yeah. Maybe someone can talk about glycogenolysis and glycogenesis simply. If there's no one and Patel, then we can just move it. there's no one willing to take us through that specifically.
I think if nobody is having it, we can Google and see it, because at the end of the day we need it. Please, if we can display it and go through the process a little to help us.
Okay, so glycogenolysis is basically converting or breaking down of glycogen into glucose molecules. So the enzyme that is involved in glycogenolysis is mainly glycogen phosphorylase, which now cleaves the glycogen polymer, because we know that glycogen is branched and it has all these glucose molecules. So it cleaves and it gives you the the glucose molecules. And then this enzyme glycogen phosphorylase, it cleaves the glycogen at glucose glycogen phosphorylate, it cleaves glucose from glycogen as glycose 1-phosphate. And then it converts it to glucose 6-phosphate, and then you have that until it's converted back to glucose. Then this glucose 6-phosphate is converted to glucose in the liver by glucose 6-phosphatase. So we usually, when we glycogenolysis is increased when like you're doing either exercise or your body is like in a fasting state, because you need that glucose, especially for your brain, because your brain just uses glucose as a source of energy. Yeah. Do you guys need me to repeat that?
Yes, please.
Okay, so glycogenolysis is from glycogen to glucose. So the first step is glycogen to glucose with the enzyme glycogen phosphorylase. And sorry, it's glucose 1-phosphate. So glycogen to glucose 1-phosphate. But the enzyme is glycogen phosphorylase. Then from glucose 1-phosphates, it's converted to glucose 6-phosphate through like a series of some other chemical reactions. Like I think it's like a long process. Again, over here you would use a kinase because you'll be adding ATP. I'm not sure about that. Just, it's okay, don't write that. I'm not too sure about that. Yeah, and then this glucose 6-phosphate is converted to glucose in the liver by glucose 6-phosphatase. Yeah.
Thank you.
Okay. Molecules that bear charge groups of opposite polarity are known as amb ions, ion conversions, zeta ions, or amphions.
Can I take you through the glycogenesis, formation of glycogen from glucose?
Yes, yes, yes.
Kindly.
Okay, from the name is the formation of glycogen from glucose, which occurs in the liver and in the skeletal muscles. And it starts from glucose, which is first phosphorylated by hexokinase to glucose 6-phosphate. Then the glucose 6-phosphate is converted to glucose 1-phosphate by phosphoglucomutase. Then the after formation of the glucose 1-phosphate, the glucose 1-phosphate will react with one molecule of uridine triphosphate to form the uridine diphosphate glucose, which will then be acted upon by the enzyme glycogen synthase to form glycogen. That's all.
Thank you so much. Okay. Question 51.
I think Zwitter ions.
Zwitter ions.
Yeah.
Yeah. Amino acids required in the human diet and not synthesized by the body are called special, trace, essential, accessory.
C. Essential.
Essential.
Yes. The membrane lipids are hydrophobic, amphiphilic, both hydrophobic and amphiphilic, or hydrophilic.
I don't know why, but I'd go with amphiphilic because I mean the head is hydrophilic and the tails are hydrophobic. I'd go with amphiphilic.
Yeah, amphiphilic is the answer.
We've talked about amino acids and that, so it's fine. The smooth endoplasmic reticulum is the site for phospholipids, amino acids, carbohydrates, or proteins.
Phospholipids.
I don't know why I would go with maybe carbohydrates because the smooth endoplasmic reticulum is mostly for steroids, lipids, and carbohydrates. But the moment we add a phosphate group, then it is packaging via the Golgi apparatus. I'm not so certain, but I would go with carbohydrates high.
Actually, the smooth endoplasmic reticulum is responsible for synthesis of phospholipids and cholesterol, like the essential lipids.
Okay.
I agree with Beldina. Majorly is the lipids. There's some synthesis of carbs in the smooth, but majorly it's phospholipids.
Thanks.
So for the proteins, it would be the rough endoplasmic reticulum. No.
Yeah.
Right? And then so
Yeah.
if you're going for carbohydrates, it would be cytoplasm.
Please repeat. Harold, please repeat.
I'm asking for phospholipids, we said smooth endoplasmic reticulum. For proteins, it would be the rough endoplasmic reticulum. So for carbohydrates, it would be the cytoplasm. Or is there like a specific organelle for carbohydrates?
Now, the smooth endoplasmic reticulum also synthesizes carbs. It synthesizes carbs, lipids, and steroids. but majorly, it majorly synthesizes the phospholipids. Right? Like the carbs are just, they are synthesized, yes, but they are not the main product of the smooth endoplasmic reticulum. So it's for carbs, I'd say it's still smooth, but not the main product there.
According to Google, plastids, these are organelles that are found in the cytoplasm of plant cells, and that is where you can have carbohydrates synthesized. But that's for the plants. The plasma membrane has selective permeability due to the presence of specific amino acids, proteins, carbohydrates, or proteins and carbohydrates.
I will go with the proteins.
Me too.
Let's go for proteins. In gel electrophoresis, what fragments will move most quickly through as gels? Large fragments, large genome, small fragments, large fragments, and large genome.
I'm thinking small fragments, but I'm not too sure.
Okay, as we are doing this, can somebody remind us of the principle of gel electrophoresis? Okay, so gel electrophoresis is just a method to separate DNA, RNA, proteins. It's based on their charge and size. So the molecules are separated using an electric field, and the molecules that travel through the pores in the gel at the speed is inversely related to the length. So this means that small molecules will travel at a greater distance through the gel than a larger one. So the answer is small fragments. Infection caused by Streptococcus pyogenes include the following except food poisoning, cellulitis, impetigo, and pharyngitis.
Food poisoning.
It's caused by E. coli.
Yeah. Mostly, I think it's E. coli that causes.
Do you want to remind us about anything special about Streptococcus pyogen?
It's just it's a gram positive cocci, and it, like, when you want to differentiate between Streptococcus and Staphylococcus. Streptococcus, like, you'll find them in like chains, while Staphylococcus will be like in clusters. Yeah, I don't
There
know
was
what
something
else.
about catalase positive and negative.
Yeah, stuff is catalase positive.
Yeah, true.
Staph is catalase positive.
Yeah.
Yeah.
Just
Then
a minute.
there's also about the hemolytic properties where
Wait. Yes.
this pyogenes is it's beta hemolytic. This means there will be full hemolysis of RBCs. Yeah.
It's also capsulated.
Capsulated.
And they're facultative anaerobes.
Then the other stuff is that it also causes necrotizing fasciitis, the flesh-eating bacteria.
That is staph, yeah?
No, strep. This strep pyogenes.
It causes
Necrotizing fasciitis.
I don't know the spelling.
And then, guys, there was something to do with group A and group B. Which one is group A?
Group A is this one, Streptococcus pyogenes. Then group B, Streptococcus, is the Streptococcus agalactiae, which causes meningitis in kids, in neonates.
Oh, yeah.
Yeah.
So you also know, in addition, that the group A, the pathogens, is also responsible for rheumatic fevers, yes.
Is this how you spell it? Agala? What?
Galaxia. Yeah. AE. Yeah, something like that. It's MCQ. You don't have to worry about it.
Happens in neonates. Huh?
Yeah, meningitis in neonates. Yeah.
We also have a group D, which is commonly for Enterococcus faecalis, commonly causing the UTI infections.
Now, also, we have the viridans streptococci that will cause the endocarditis, infective endocarditis.
In terms of like, if you want to culture, which media will you use? Blood egg because of hemolysis. I'm not sure.
Let me check and see if I get it right now.
It media with blood so that it can allow the detection of beta hemolysis.
Yeah, blood agar, yeah.
Do we need to remember the colors around the colonies for alpha, beta, and gamma? Just I'm seeing here that alpha will have green rings around the colonies. I don't know if it's necessary, but beta.
Oh, yeah, it's necessary. Yeah, beta will be clear because all the cells have been lysed. Alpha will be green because of that partial hemolysis.
Gamma will be no color change.
Yeah.
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 Okay, that was nice Okay.
That was comprehensive.
Yes, thank you. The following have cell wall except Mycoplasma pneumonia, Escherichia coli, and gonorrhea and S. aureus.
For Mycoplasma, because it's an atypical organism.
Yeah. Should we just do like the other one, recall?
Pardon?
Should we talk something about pneumonia, Mycoplasma pneumonia?
Yeah. I'm sure. I'm seeing people microbiology. They are very well prepared. I think we should give them a chance to feel yes.
I'm not sure, but somebody can confirm. For Mycoplasma pneumonia, I think it causes like hospital-acquired pneumonia or atypical pneumonia.
Yeah, it causes hospital-acquired.
And then it's the streptococcus that causes now the community-acquired.
Yes.
Yeah.
You said strept. No.
Yeah, streptococcus pneumonia.
Yeah.
It's the one that causes community.
Maybe still an little addition on the organism lacking the cell defective of the cell wall. As you said, we have Mycoplasma. We also have Rickettsia and Chlamydia. And remember that Chlamydia, we have Chlamydia trachomatis, usually commonly causing some conjunctivitis in children and also the UTIs. So apart from Mycoplasma pneumonia, we also have Rickettsia and we have Chlamydia.
Which one of the following is transmitted through droplets? Cholera, bacterial meningitis, malaria, or syphilis?
I think it's bacterial meningitis. But I've ever had a patient try to convince me that they got syphilis through coffee. I don't know. He was really convinced. The patient was shy, to be clear. Yeah, so it's bacterial meningitis because of the organisms that cause it. Strep pneumoniae, Haemophilus influenzae. Those are organisms that are transmitted through droplets. Then for cholera, so you know cholera is a fecal oral route. Oh yeah, so here majorly contaminated water, contaminated food. Yeah, malaria is mosquitoes, syphilis is a STD, but you can put in 0.01% of cases. Then you put into brackets, chai. It's transmitted
Chai.
through droplets. Yeah, this guy. But he got
So for
better.
the
That's what matters.
So for the causative agents, we'll be talking on Vibrio cholerae for cholera, on bacterial meningitis, as Spider-Man also indicated. Also remember you have Neisseria meningitidis. Under the Neisseria we have Neisseria gonorrhoeae and Neisseria meningitidis. But meningitis is specifically for mostly causing the bacterial meningitis. For malaria, we'll talk it under protozoa. The first param is a protozoa. Then the syphilis will be a sporocrites, that is Treponema pallidum.
And then just to remind us of something we had said about Neisseria meningitidis, it's the one that causes the petechial rash. It's a unique, in meningitis, we know the classic triad, the headache, fever, and equal rigidity. But specifically for Neisseria meningitidis, we'll see a petechial rash. That's in the torso region, especially in the torso region or in the hands, we'll see pinpoint bleeding. That kind of narrows it down to Neisseria meningitidis, yeah.
Sorry, could you please remind us of the triad between for the bacterium meningitis? Fever?
Pardon? Oh,
triad.
fever, headache, and neck rigidity or knuckle rigidity.
Okay, that's good. Do you want to cover the treatment?
Now for treatment, it will depend on the organism, but essentially because first of all they start a broad spectrum before they can identify the organism. They'll give a beta-lactam and an aminoglycoside. So usually something like ceftriaxone or cefotaxime with gentamicin. Yeah. Then once they narrow it down to a specific organism with the specific sensitivity report, they will now give the specific drug, yeah.
Guys, Abu pronounce this thing.
Yersinia pestis.
That one, yeah. Is a gram-positive bacteria, is motile, causes bubonic plague, and is spread by mosquitoes.
That one is, it causes the plague. Was it gram negative? Hopkins.
Do you want to add anything about?
Gram-negative rod. Coagulase positive. Also causes pneumonic plague.
Sorry?
Also causes pneumonic plague.
Stand by, please.
Hi, we continue from here, Shrina, tomorrow. It's already
Yes,
10.
okay, Fatma. Okay, Fatma. Okay.
Thank you. Thank you, guys. Thank you all. Let's
Thank
make it
you.
productive like today. It was very insightful and very educative. Thank you all. Have a good night.
You too.
All the best, guys.